Okay, good afternoon. Sayed, there's no one in the session on you, so I guess let's start and then. Oh, hello, Angela. So I guess others are gonna be watching the recorded session. Okay. So today we're gonna continue the course of biology, and we're gonna start speaking about enzymes. So, one second. Okay. So what are enzymes? Enzymes are biological catalysts. Okay, Angela is asking. The question yesterday, Grant, what the title of the course to watch. Issues. So they are biological catalysts, and usually what does catalyst do? Catalyst usually speeds up the reaction. So their function is to speed up the reaction. Okay. However, these enzymes are not consumed during the reaction. What did I write? Okay, consumed during the reaction. Thus, they are degenerated at the end. Okay. Enzymes can be proteins and they. So to be able to, how does our body work? Let's speak about this. Our body needs something to speed up reactions inside. Okay. However, our body's temperature is a constant uh, characteristic in our body, which is 37 degrees Celsius, which our body itself, if it wants to increase or to accelerate the reaction, what happens? It cannot change the body temperature. So since usually the usual chemical reactions that happen in the laboratory, we increase the temperature for them to accelerate. However, our body's temperature is constant. We cannot use it. Other than this, we cannot increase any reaction in our body in a mechanical manner. So we end up having only enzymes is the chemical thing that helps in increasing body reactions. So other function for the enzymes is they facilitate the formation of the product, but they does not change the product. If there was enzymes that enter the reaction in our body, they facilitate the formation of this product. They make it easier to form, faster to form, but the product is not changed as an identity. Okay. Now, enzymes usually are specific for their substrate Okay. And the enzyme rate usually in the body is from 10 power 5 to 10 power 7. This is the rate. Okay. Okay, now let's take a graph. Let's draw this graph. So here we're gonna have, let's put, okay, let's name them letters. Let's name this letter R, this letter P. Okay, perfect. So if you look here in the figure, this figure which is up, or this curve which is up, is without enzyme, is a reaction without enzyme in the body, and the one which is down is this one, is with enzyme. Okay, if you see that the one with enzyme, which is this, has an activation energy, the inside is the activation energy. Okay, so the one with enzyme has this activation energy, and the one without enzyme gonna have all of this activation energy. Okay, so the activation energy with the one with enzyme is reduced, so it decreases the activation energy. However, if you see that it reaches the one with enzyme, it reaches the top faster and it goes down faster. It reaches from we go from the reactant to the product, we're gonna reach the plateau faster, which is up, and then we're gonna reach down super fast. So it's faster, so it increases the rate and decreases the activation energy. Here is the activation energy. It's much less than this. This all is the activation energy of the one without enzyme. This is the activation energy with the one with enzyme. So it decreases the activation energy and it increases the rate or the speed. Okay. Now here, the highest point on the graph is called the transition state or the plateau. Okay. What happens on this transition state? What happens on this plateau? All the active sites are occupied. This means that the active sites of the enzymes are now all occupied when it reaches a plateau. So plateau means when all active sites are occupied. Okay. Okay. What is delta G? When we drew the graph, we drew it like this. Okay. Delta G, as a definition, okay, it's the amount of energy needed to convert 1 mole of reactant from ground state to transition state. So we have here the reactant, here the product. To convert 1 mole of this reactant from this state from here to reach the transition state, this is the delta G. It's the amount of energy, how much energy we need to convert this amount from the ground state to the transition state. Yeah. So the enzyme, they increase the rate or the speed and they decrease the activation energy. How does the enzyme look like? This is the enzyme. And here, okay, here we have the substrate. Here is the two active sites. When these two active sites are fully occupied, the graph reaches the plateau. Okay, you understood how? Okay, so, okay, perfect. So here we have mono, here we have mono, like one, another mono and mono together, they will give you a di. Okay, so the shape of the enzyme, it's a kind of receptor-like looking, and the substrate will gonna compete or gonna sit on this active site competing with other substrates depending on the shape of the head of the substrate and the shape of the active site of the enzyme. Okay, okay, perfect. Before I continue, or let's leave this for when we speak about genetics. Okay. Okay. Now we have to speak about enzyme inhibition. Enzyme inhibition. So of course we have the covalent enzyme inhibition and the non-covalent enzyme binding inhibition. Okay. Means we have the reversible enzyme inhibition. However, for the Imat, you're asked about the non-covalent and which is the reversible. And the covalent is the irreversible. Non-covalent is the reversible. For the Imat, we're asked, we have to focus about this, okay, more than the other. So let's start with the non-covalent or the reversible. Okay. So let me see the chat. Okay. 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 So types of inhibitors, or let's first speak about what are inhibitors before we continue. So inhibitors are compounds that bind to enzymes and decrease the enzymatic activity. As an example, like drugs, like toxins, and etc. Okay. Types of inhibitors. Okay. We have four types. We have reversible, non-competitive, uncompetitive, allostatic enzymes. Now let's speak about the reversible. These kind of enzymes, okay, or let's say, yeah, these ones, they bind covalently, means they share between them electrons. In order to bind, how does the enzyme look like? As we said, the enzyme looks like this. So since the enzyme has an active site, on this active site, there are so much molecules are competing together to be able to take this active site of each enzyme. The most molecule which looks the best in shape and affinity, it will be able to succeed in sitting on this active site of the enzyme. Okay? Now, now, uh, now we have to speak about each one of them. Let's start with the reversible immediately. So, the reversible inhibitors, these they have the substrate structure, the same structure of the substrate. Okay, so since they have the same affinity of the substrate, what happens to them? They bind on the active site, competing with the substrate. So it looks like this. Here we have the enzyme. Okay, here it's non-covalently bonded. Here we have the inhibitor. Okay, perfect. So when the inhibitor binds to the enzyme, what happens? No reaction will happen. Why no reaction? Because it's an inhibitor. It stops this work of the enzyme. So then the reaction will stop. However, if the substrate was able to succeed in binding inside instead of the inhibitor, the reaction will occur. Okay. Here we have, or we speak about this later. Let's continue the perfect. Now we have to speak about the non-competitive inhibitors. Non-competitive inhibitors. So we have the inhibitor or the substrate binds simultaneously to the enzyme. Okay, so binds now. We have two kinds of these non-competitive inhibitors. The first kind is called the pure inhibitor, and the second kind is called the mixed inhibitor. So we have the pure inhibitor, which is the non-competitive pure inhibitor, and we have the mixed inhibitor. The pure inhibitor, what is its definition? As a definition, it binds away the active site and changes enzymes conformation. Okay. However, the mixed inhibitor, it binds close the active site and affects the affinity of the enzyme for the substrate, means it decreases substrate affinity. So, as we said that the substrate and the inhibitor will be competing to be able to bind to this enzyme. Let's say we have this enzyme. If we have a pure non-competitive inhibitor, it will come, here is the active site, okay? It will come and bind far from the active site, here or here or here, okay? And when it binds, it will immediately give a sign for this enzyme that I bind it, and then this enzyme will change its conformation. So it will become, let's say, in this shape. And when it becomes this shape, it might make its conformation looking the same way it looks like, because, and then it will be able easily to come and sit here. So this is a non-competitive inhibitor because it's not competing with the substrate. It's just trying to change this enzyme to look like how it looks like. Okay. However, the mixed inhibitor, which is, let's say we have this enzyme, it binds close to the active site, let's say here, okay, and affects the affinity of the enzyme for the substrate. What does it do? It decreases the affinity. It tries to not allow the substrate or the enzyme to accept shapes that look like the other substrate. Means it does not change the conformation as the pure one. However, it tries to decrease the affinity, the probability of this enzyme to accept the substrate looking like the active site. So here it decreases the affinity. However, here it changes the conformation of the enzyme. Okay. The one of changing conformation, it binds far from the active site. The one of the mixed one, it binds next to the active site. However, which one of these two is the more effective? It's the mixed inhibitor. So this one is high effectivity, works more. However, the pure inhibitor is less effective. Okay, now let's speak about the uncompetitive inhibitors. So inhibitors bind to the enzyme substrate complex ESC, no reaction. How does the uncompetitive inhibitor work? It so let's say we have an enzyme, okay? And we have a substrate sitting on the enzyme normally, and the reaction is happening and is being catalyzed, and everything is going well. This uncompetitive inhibitor will come and sit here. So it binded not to the enzyme without the substrate. It waits for the substrate to sit on the enzyme. After it sits on the enzymes, it comes and it inhibits the whole complex. So now no reaction, it inhibits the whole complex. If you remember the first one, which is the competitive inhibitor, they used to not allow the substrate to sit. They come and sit instead of the substrate on the active site. However, the uncompetitive inhibitor, they wait for the substrate to sit and then they sit next to them and they stop them from. Okay. Now the last one is the allostatic enzyme. It resembles the substrate. It stops the substrate. It comes, yeah. The competitive ones resembles the substrate. Yeah. If you mean the competitive, the first one, yeah, they come and sit on the enzyme on the active site as if they are the substrate. Try to resemble the substrate for the enzyme to stop working, to inhibit the enzyme. Okay. Now the allostatic enzymes. So there are two types. We have the first type is inhibitor, allostatic enzyme. The second type is the activator. Let me draw a chart. So okay, this is the activator chart, allostatic enzyme. This one is the inhibitor allostatic enzyme. And this is the control, the non-effector. We call it the non-effector. We call it usually the control. Okay, means the normal one without the allostatic enzyme. Okay, so number one thing you should know about the allostatic enzymes that they have an active site usually, like any other enzyme. Okay, an active site plus one regulatory site. Means they look like this. Let's say this is the enzyme. They have here, this is called the regulatory site. And here is the active site. Perfect. So the activator, if you see, it activates. Here is the normal graph, okay? If you see that the graph was moving in a way which is constant and not increasing, activator, it increases this work of the enzyme dysfunction. However, the inhibitor, it inhibits and it goes down the non-effector or the normal graph. Now let's speak more about the activator and the inhibitor. So the activator, the definition of the activator, allostatic enzyme. So activator Okay. So the activator allosteric enzyme, allosteric inhibitor, I mean, not enzyme. No, here we call it the allosteric activator. This is the name. Allosteric activator. So after binding, it increases the affinity of the enzyme for the substrate. This means it tries to activate the enzyme more. It's not an inhibitor. This allosteric enzyme, this allosteric activator, it sits on the enzyme and it tries to allow or to increase the affinity or the probability for the enzyme to be able to bind easily for the substrate, for the normal substrate. So this it increases, it has a very good effect on the reaction. However, the inhibitors, so the allosteric inhibitor, after binding, it changes the conformation of enzyme for substrate. So the allosteric activator, after binding, it increases the affinity of the enzyme for the substrate. It makes it easy. However, the inhibitor, it changes the whole conformation of the enzyme or the active site of the enzyme for the substrate not to be able to sit on. Okay. So we spoke about the different kinds of inhibitors that we have, the allosteric enzymes, and all of it. Now, before we continue, I want to give a formula, okay, and I want to explain something about it. So the formula for so when the enzyme is saturated means when it's fully equipped. So when the enzyme V0 means the speed equals V maximum times the times or times the concentration of the substrate over Km plus the concentration of the substrate. Km is the constant. Okay, so now according to this formula, we can know each kind of inhibitor, what happens according to this formula, what happens to the inhibitor. Okay, so let's speak about different types of inhibitors. We spoke about them. Is the reversible inhibitor. We have the non-competitive. We have the uncompetitive. And we have, no, the allosteric is an enzyme. Okay. So reversible inhibitors usually have high Km. So when Km increases according to this formula, V maximum is constant. This for memorizing. Each one of them is for memorizing. Non-competitive has a Km which is constant, and the V maximum is low. And the uncompetitive has the Km low and the V maximum low. Okay, so all you have to memorize is the formula, because they might give you the concentration of the substrate and ask you to get the V maximum, or they might give you this formula without you memorizing it, and they will tell you according to the reversible inhibitor that we have here. Tell me about the Km and the V maximum. The options will be like Km high and V maximum constant, Km low, V maximum low. So I suggest memorize them and go, because how are these made? How did they reach or conclude here? They worked as physics, like directly proportional, indirectly proportional, until they reach this conclusion. So try to memorize them, is much easier than wasting time on trying to understand during the exam each one of them. Okay. Now, here only when the enzyme is saturated, we use this formula. Okay. So what changes the conformation of the enzyme are the inhibitor allosteric enzyme and the pure inhibitor of the non-competitive inhibitors. What changes the affinity are the mixed non-competitive inhibitors and the activators. The activators change the affinity positively by making it more loving the substrate. However, the non-competitive mixed inhibitor, they try to change the affinity in a negative way by decreasing it and not allowing the substrate to bind to this active site. Okay. So about the enzymes, I guess we're done. This is everything you should know for the Imat about it. Okay, perfect. So now I want to speak about a note, just like a small note before it does not do nothing with the enzymes, but I'll say it. So out of 600, no, yeah, out of 600 subunits, only 200 are bases. This means 1/3 of 600 subunits are bases. Means 1/3 of the subunits are bases. So if they got you in the question, okay, let's say they got you in the question, we have, let's say, 400 subunits, and how many of these are bases? You should know that you just divided by 3 to end up with the bases. Or even if they ask about something else than the bases, if there's in the question something in a way that you should let the bases interfere in your answer, so it's a kind of trick. You should, before you continue answering, you should know that 1/3 of them are bases. Before you do any replication or mutation or whatever, you should make sure that you calculated the bases correct and you didn't take 600 bases. You should know that you should take the third of them to be bases, because this also might be a kind of trick in the IMAT. Okay. Okay, perfect. So, I don't think they sent you the PDF. There's a PDF that we checked everything we already did and what we still have to do. So, Let me check. So we spoke about enzymes. Enzymes is done. Mhm. Prokaryotic DNA, we spoke about it. Eukaryotic DNA, we spoke about it. Still have genetics. And yeah. So before we start genetics, I want us to start on something else, which is, let me see chat. The nitrogenous bases that we have, they might tell you, let's say, calculate, let's say they use restriction enzymes. Let's say the question is this. They might use restriction enzymes and tell you how many base units it made. So here, to be able to calculate, they might give you in the question, we have, let's say, 600 subunits of the DNA, and we cutted it in this part, you know. So when they say we cut it in this part, you cannot calculate the subunits only if you know the number of bases. But you should know that out of this 600, we don't have 600 bases. We have 200 bases, because not each subunit has the same amount of bases. And it does not mean that it can be just the question about restriction enzymes. It can be mutation. It can be so much things that do with the nitrogenous base. Okay, I will try to find the question that came like this, and we try to solve it. Don't worry. So now, now we will speak about skeletal system. So skeletal system, we have, it's divided into axial bones and appendicular bones. So we have the axial part and the appendicular part. Axial, everything inside; appendicular, everything which is shown to the outside of the body. So appendicular part. We have 80 bones which are axial in the body, and we have 126 bones which are appendicular in the body. So if you add 80 plus 126, we're gonna end up with 206 bones as a total in our body. Okay? The axial ones are the skull, 29. This for memorizing. The thoracic cage, 25. The vertebral column, 26. Okay? The appendicular are, we have 2 arms, 2 legs, the clavicle, which is under the neck part, the scapula, 2 hands, 2 feet. Now in each one, of course, we have so much bones, so in total there are 126. Okay? So let's speak about the vertebra or vertebral column, which is so important for the Imat. So the vertebrae. We start out like this, looks like this. Yeah. So we have, here is the cervical part. Here is the thoracic part. Here is the lumbar part, coccyx. Here we're reaching down, and then the sacrum. So, cervical, thoracic, lumbar, coccyx, sacrum, seat, CTLCS. Okay? Cervical, we're gonna have 7 bones. Thoracic, we have 12. Lumbar, we have 5. Coccyx, we have 4. But all of these 4 are fused in one. So they are fused. Like if you look at them from outside of the coccyx, you'll be like, okay, this is one bone. But if you come closer, it's 4 bones. So they are fused together. And the sacrum is also 5 fused together. Also fused. Okay. However, now the bone which is between the neck, above the neck, and the vertebral column, this one here, is called the atlanta. Okay, from where the word Atlanta came, because here we have the C1, the very most number 1 bone of the vertebra is called the C1. Why do we name C1? Because cervical one, we start with the same one. Okay, so we have the very first 2 bones are C1 and C2. C1 is the atlas, very first bone, and the C2 is the axis. So the bone, which is the very first bone between the neck, between the backbone between the neck and the vertebrae, is called the atlanta or the atlas. Okay. What does the atlas have or composed of? The neck, plus skull. Okay. Plus skull. Okay. And 12 cervical. However, the axis is the very most bone which is under the atlas or after the atlas. Okay. Okay, let me see chat. Isn't coccyx 4 and sacrum 5? Yeah, coccyx 4, sacrum 5. Okay. So, what else? Perfect. Now, what you should know for the Imat is just these. Now, before I continue, I want to show you just how it looks like, the sacrum and the coccyx. So we have the coccyx from the outside. You have the sacrum from the inside. Here is the sacrum, which are 5 bones, look like one. If you see, it looks like one. And here is the coccyx. If you see, it also looks like one, but it's usually 4 bones together. Okay, this when you reach the end of the vertebra. Okay, now I want to give you some biological words that might help you. So we might use superior, inferior, ventral, or anterior, dorsal, or posterior, axial, or proximal, appendicular, or distal, lateral, or medial. So superior means up, inferior means from down. Ventral or anterior means from the front. Dorsal or posterior means from the back. Okay. Axial or proximal means from the inside. Appendicular or distal means from the outside. And lateral means medial from the middle. These are important because these are names that might help you because most of the anatomy names are these are used. Okay. Now we're gonna speak about the brain anatomy. Brain looks like this. Okay. So here we have the frontal lobe. Here we have the parietal lobe. Here the occipital lobe. Here we have the cerebellum. Here is the temporal lobe. Here goes the spinal cord. Okay. Perfect. Frontal is from the front. Parietal is from the high from up. Occipital is from inside. Cerebellum is from the back. Spinal cord also comes in the back, and then the temporal lobe is from the bottom. Okay. So, what does each one of these do? So, each one of them has a function. Let's start with the frontal lobe. Frontal lobe helps in motor control, problem solving, and speech. Temporal lobe helps in auditory function, means hearing, language, and memory. Cerebellum helps in balance, coordination. Occipital lobe from its name and the side, and the brain stem, like the medulla oblongata. It helps in metabolism, like digestion, like heartbeats, like breathing, etc. Okay. Perfect. Okay. Now let's continue. We didn't speak about one of them. Yeah, the parietal lobe we didn't speak about. So the parietal lobe it helps in sensory body orientation or sensation. So sensory body orientation. Okay. Now we continue speaking about the brain anatomy. So let me try to draw it. So is the brain. We're still in the brain anatomy part. So we have here. So first we have here a layer. This liquid layer is called the meninges. Meninges. We have here the brainstem that we said it helps in digestion and metabolism. We have here the spinal cord. We have here the thalamus. We have the corpus callosum here. We have the bottom, the hypothalamus. And then down here we have the pituitary gland. Here we have the pons. Here is the medulla oblongata. And here we have the cerebral cortex. Yeah. So what does each one of them do? The cerebral cortex, it has perception, memory, attention, language, consciousness. Let's start writing them. So, cerebral cortex. Attention. Why the handwriting? 1 second. Okay. So we have attention, memory, perception, language, consciousness, and what else? Sensory processing, motor control, these are the cerebral cortex. Okay. Now we have the white matter. It's the white matter is right under the cerebral cortex. So the white matter is right under the cortex. Okay, it consists of myelinated axons that facilitates communication between different regions of the brain. Okay, so who else than the white matter helps in the coordination and I mean the communication is also the corpus callosum. So both of them, the white matter is the and the corpus callosum. Um, we have the basal ganglia. It's a group of subcortical structures located deep within the brain. It is involved in motor control, like learning, habit formation, like routine, structures that it includes, so structures that the basal ganglia includes, are the striatum, the globus pallidus, substantia nigra, and the subthalamic nucleus. Okay. Now, we have the thalamus. It's the relay center or the relay station for sensory and motor info to the cerebral cortex. So it's the relay center for sensory and motor information to the cerebral cortex. It plays a role in regulating consciousness or conscious, sleep, and even alertness. When the body is on alert, the thalamus is working. Okay, now the hypothalamus. It regulates homeostatic process like temperature, hunger, so it regulates homeostasis and even hormones. Let me write on the paper that I have to teach you something about hormones for I don't forget before we end the course. So, hormones. Okay, so it regulates homeostasis processes like temperature, like hunger, like thirst, like sleep-wake cycles. All of this is the hypothalamus. It serves also a link between the nervous system and the endocrine system. Endocrine, which produces hormones in blood. Okay, perfect. Now we have the brain stem. It's composed of midbrain, of pons, and of medulla oblongata. As you said, it works on respiration, heart rate, metabolism, blood pressure, digestion, and it facilitates the communication between the cerebrum and the rest of the body. Okay, now the cerebellum. As we said, it's for coordination, spoke about it, and emotional and cognitive process. Okay, and at the end we have the ventricles, which are the fluid-filled cavities within the brain. They produce and circulate cerebrospinal fluid, which cushions and supports the brain. Okay, now before we continue about this, before we leave the brain part and we start with another thing, I want to speak about three other things which compose also the brain. Okay, so let me see how many participants we have. Saad, hello Saad. Okay, now want to speak about the different tissue layers or membranes that make up the brain. So, tissue membranes that makes up the brain. They are 5 layers. We start with the first layer or the outermost layer, which is called the dura mater. So the one is the dura mater. So as a definition, this dura mater is the outermost, the toughest. Why it's the toughest? Because it's the main protection of the brain and the most resistant membrane surrounding the brain. It's also known as the tough mother membrane. It provides a tough or thick protective covering for the brain. Number 2, we have the arachnoid matter. The arachnoid matter is the middle layer. It's located between the dura mater and the pia mater. It contains cerebrospinal fluid in the subarachnoid space. It also helps in protection. Okay, so dura mater, under the dura mater we have the arachnoid matter. I'm good, how are you, Sad? Okay, now we have third is the pia mater. So it's the innermost delicate membrane that closely follows the contour of the brain. It provides nourishment and support to the brain tissue. It closely adheres or sticks to the surface of the brain. Okay, so we have dura mater, then arachnoid matter, and then the pia matter. They are in this sequence. This thick membrane, this one is the middle one, and the last one is very thin membrane. Okay. Now we still have two. Cerebrospinal fluid, which are in the ventricles that we spoke about. If you remember, we were speaking about that we have ventricles in the brain anatomy. These ventricles gonna hold the cerebrospinal fluid. So the cerebrospinal fluid is a kind of protection for the brain, like any other tissue that we spoke about. So cerebral fluid, CSF. It's the fluid that fills the ventricles and the subarachnoid space. Okay, it provides cushioning and buoyancy to the brain, as well as it facilitates nutrient delivery and waste removal of the brain cells. Okay. The last one is the blood brain barrier. It needs a full page, so a highly selective semi-permeable membrane that separates the circulating blood from the brain extracellular fluid. So since, as we said, that the brain has an extracellular fluid, if if this extracellular fluid that existing in the blood and the brain mixed with the blood, it won't be able to do its function correctly. So we need a membrane that separates the blood of the body, the circulating blood in the body, which is the only liquid that transports in the body from this liquid of the brain, because the liquid of the brain must stay far from blood. So it's the blood-brain barrier. It's a kind of tissue or membrane, just super selective to separate them. What's its function? Its function is protects the brain from harmful substances while allowing essential nutrients to pass through. Okay. So this is mainly everything about brain you should know. Okay. Okay. So we still have the body systems. We did the digestive, we did the skeletal, we did the muscle part. We still have immuno, respiratory, digestive. No, digestive we did. So immuno, respiratory, urinary, and reproductive. Immuno, respiratory, urinary, reproductive. There's still one. One second, let me check. Nervous. Skeletal, muscular, we're done. We have to speak about the endocrine system or the hormone. And let me just write about a system which you just should know a small detail about it, nothing else, which is the integumentary system. So okay. So, for the skin, we can call it. Since the skin is an organ, it's not a tissue. The skin is an organ. It's the biggest organ of our body. Before we continue, I want to ask the question. The question is, which kind of cells in our body that has the most mitochondria? What do you think? Cardiac, bravo, Sayed. So the cardiac muscle cells has the most mitochondria. Okay. So what are the functions of this integumentary system? It helps in protection, sensation, thermoregulation, vitamin D synthesis, immune function or immunity. How is the skin divided? Or what is it made up of? Or this integumentary system, usually what's made up of? It's made up of the skin, of the hair. This all inside the integumentary system. The nails, the sweat glands, the sebaceous glands. You're not really asked about them. You might just be asked about the skin. The skin is made up of 3 layers. Looks like this. It has the epidermis. So the skin is the outermost layer of the integumentary system. Is the highest layer of the integumentary system, and is composed of 3 main layers. We have the epidermis. We have the dermis and the hypodermis. Okay. The epidermis is the outermost layer of the skin and consists of different types of cells, including keratinocytes, melanocytes, Langerhans cells. We already spoke about these, and we already spoke about them in detail before. Okay. The dermis is the middle layer of the skin and contains blood vessels, contains nerves, hair follicles, and sweat glands. So blood vessels. So the blood vessels are not in the epidermis, are in the hypodermis and the dermis. Okay, does not reach up. So the dermis is the middle layer of the skin and contains blood vessels, nerves, hair follicles, and sweat glands. Here start the hair follicles and they go up to show the hair from the dermis and not from the epidermis. Okay, and the hypodermis is the innermost layer of the skin and is made up of adipose tissue or the fat tissue, which provides insulation and cushioning for the body. The hair, let's speak about the hair, since we already speak about the integumentary system. The hair is a keratinized structure, okay, that emerges from the skin and serves various functions such as insulation, sensory perception, and protection. Hair follicles are structures in the skin that produce and anchor the hair. The color of hair is determined by the amount and type of melanin pigment produced by specialized cells called melanocytes. The nails are hardened, protective structures that cover the upper part of the fingers and toes. Nails are made up of keratin and they play a role in protecting the underlying skin and enhancing the sensitivity of the fingertip center. Since we have so much pain receptors on the nail parts, on the beginning of the fingers parts, so this nails try to protect, okay, and help in the sensitivity of this finger. Sweat glands are specialized structures that produce and secrete sweat, a watery substance that helps regulate body temperature. There are two main types of sweat glands. We have the eccrine and the apocrine. Let's write about this. So, sweat glands. We said that they are specialized structures that produce sweat. So, they are made up of eccrine sweat glands or the apocrine sweat glands. The eccrine sweat glands are distributed, so let's write here, are distributed over the entire body and are responsible for most body sweat. The the apocrine are found in specific areas such as armpits, groin, and are associated with odor. So the apocrine are specialized and the eccrine are the non-specialized ones for the whole body. Okay? And the last one of the integumentary system is the sebaceous glands. Sebaceous glands are oil-producing glands located in the skin. Primarily, these sebaceous glands are produced from the face, the scalp, and other areas of the body. These glands secrete sebum. So, let's write sebaceous glands. They secrete the sebum. What is the sebum? It's an oily skin substance. What's its function? Okay, helps maintain the skin's barrier function and it prevents water loss. Why it prevents water loss? It's an oily substance. This means that it's an hydrophobic substance. When the skin is full of hydrophobicity, this means that water will not try to go out of the skin because the skin is all of it is hydrophobic. So when water tries, it will go back inside because it's hydrophilic or it's polar. Oily substance is non-polar. So it won't attract it to go out. So that's why it helps in preventing water loss. Sebaceous gland. Perfect. Now, integumentary system is done. We still have good time to explain another system. Okay, let's speak about which system is the easiest. Respiratory. Let's try to finish the respiratory now. So, The respiratory system is so easy. We have something inside this respiratory system called the diffusion. We all know what's the diffusion. It's the passive transport in our body. And because of diffusion, we have the fusion of the exchange of the gases or respiratory gases, which are the oxygen and the carbon dioxide during the cycle. So, okay. And yesterday we forgot, before we start in the respiratory, before we start in the respiratory, I want to finish something we didn't finish yesterday. We forgot to finish. We were speaking yesterday about the muscle fibers, if you remember. We spoke about the muscle tissues, skeletal, cardiac, smooth, if you remember. So today I just want to try to draw the anatomy. Mhm. So muscle fibers. Before we start with the respiratory. So it looks like this. This is the normal muscle. Like this. Muscle fiber. Okay. One of them here, one of these circles is called the fascicle. The ones inside this fascicle, the fascicle, if you zoom in the fascicle, looks like this. So the one inside looks like this. This one here is the blood vessels. The outermost one is called the perimysium. The innermost middle one is called the endomysium. Here we have muscle fibers. Here is the epimysium. And this is the tendon. What are tendons? Tendons are the ones that allow the connection between this muscle and the bone. Okay. Any info about that for the diagnosis? We provide them whenever you guys can to make that. Logical reasoning. Guys, one moment because the door is ringing. Okay, give me 1 second. Yes, someone came. Just have to make sure that's fine. Okay, I'm sorry, guys. I have to go. So we have the perimysium, the endomysium, the epimysium, and the tendon. For the logical reasoning part, I guess Sam was speaking about it today morning. I remember when I spoke to him. He said he's preparing them. I'm at simulators. We're gonna solve together from them by the end of the course. So since I remember that we started from 5 July. 5 July? Yeah. July. No, are we in July? Start in August. Wait, okay. Started 5 August. So we should not start. We should not finish by the 30th of August. We have to stay until the 5th of September together. So like, don't worry. We still have like 2 weeks to practice and everything. Don't worry. Like we won't end the session on the 30th. We have like until 5 September. So we will have good time to practice. Okay. So okay. So we spoke about this. Okay, now we have to speak about the types of muscle fibers. So let's try to draw it short. Type, we have type 1, type 2A, type 2X. Here we have the contraction time, oxidative capacity, diameter, resistance fatigue, and generating force. Usually type 2A is super slow, have a very slow contraction time, okay? And has a very small diameter, type 1 muscle fiber. And type 1 muscle fiber has a very small generating force. So it's negative in 3 things: in contraction time, in diameter, and in generating force. However, it's positive in oxidative capacity, so it's very high, has a very high oxidative capacity, and has a very high resistance to fatigue or exhausting or tiring. Type 2A is, okay, so has a very, not a very fast, has a fast or moderate contraction time, so it's fast, has a medium diameter. Everything is moderate, to be honest, for type 2A. It's moderate, has moderate or medium diameter also, medium oxidative capacity. Now let's write here high oxidative capacity. It's better than to say medium. So high. So it's fast, it's high oxidative capacity, has a medium diameter, has moderate resistance to fatigue, and has a moderate generating force. Type 2X is super fast, very fast contraction time. The time to contract is super fast. Has a low oxidative capacity, has a large diameter, has a small resistance to fatigue, or let's say low, cannot say small, low resistance fatigue, and have a very high resistance generating force. Okay, so this also for memorizing. Perfect. Now we have to start respiratory system. So respiratory system. So how it starts? We first respire. We have it goes on two levels. So two levels. We have the alveoli or the lung blood vessel and blood vessel organ or tissue or cell. Okay. Now let's start with the first one, then we go to the second one. So the alveoli or the lung blood alveoli looks like this. The blood vessel here. This is the blood entering. This is the blood leaving. So usually the air that we respire is full of oxygen and low amount of CO2. So here we have high oxygen and low CO2. When we inspire, okay, so the blood entering the alveoli is coming from where? It's coming from the organs, right? So since it's coming from other organs and tissues, this means that it has so much waste. So it has a high carbon dioxide and the low oxygen. So what happens here? We have here a high oxygen and here we have a low oxygen. So here we have high concentration medium, here we have low concentration medium. And we always compare oxygen with oxygen, carbon dioxide with carbon dioxide, not carbon dioxide with oxygen or not oxygen with carbon dioxide. Each element is compared by itself. Okay, so high oxygen, it goes usually diffusion happens from high concentration medium to low concentration medium. So it goes here, the oxygen from the alveoli to the blood. So here, because here we have high concentration. However, the blood entering is full of CO2. So here the high concentration of CO2, and here we have low concentration of CO2. So it will start going from here to here, the CO2. Then what happens? The blood leaving will be highly oxygenated, lowly deoxygenated, and by the end, the alveoli will be full of deoxygenated air and low oxygen. And then by exhalation, it will take this carbon dioxide and get rid of it. Perfect. However, on the second side, here we have the organ. Let's say mitochondrial cell or any organ. And here the blood vessel. Here blood entering the organ, here blood leaving the organ. So the blood leaving the lungs is the same of the blood entering the organ, right? So let's name this A. Here also it's A. So it was full of oxygen and low CO2. However, when the organ respires, let's say the mitochondria, it produces so much heat and so much. And when combustion reactions happen, let's say in the mitochondria or in any other cell, after it respired, it will be full of carbon dioxide and low O2. So here we have high CO2 in the blood and no, we have high O2 in the blood and low O2 in the organ. So O2 will go from the blood to the organ. We have high CO2 in the organ and low CO2 in the blood, so CO2 will diffuse from the organ to the blood. So then the blood leaving as a product will be having high CO2 and low O2. The blood leaving here is the same of the blood entering the lung, and then it will go to the lung to be able to clean itself again and fill itself again with O2 to go to another organ. So here, let's name this B. This is also B. So blood entering the lungs is the same of blood leaving the organ, and blood entering the organ is the same of blood leaving the lung. Okay, this is respiration. There's a diagram that you have to draw before we finish respiration, and will be done for today. Okay, so the respiration diagram that you should know is or spirogram. Lung volume, to 2600, can be 3100, 4000, 5200. So 2600 is the inspiratory reserve volume. So the volume of the lung, 2600, it's the inspiratory reserve volume. 3100 is the tidal volume. 4000 is the expiratory reserve volume. And 5200 is the residual or the left volume. If you want to draw it as a chart, it looks like this. Okay. If you want to divide the chart from here to here means this part. This is the vital capacity or the vital volume that of lung volume. So the vital capacity. And here we have the functional residual capacity. However, all of this from up to down is the total lung capacity. So all you should know is the numbers and the name or the kind of volume. 2600 is the inspiratory. No, you should not know the formula. You should just know the number and what does it mean. The inspiratory volume is 2600. 3100 is the tidal volume. 4000 is the expiratory volume. 5200 is the residual volume. This spirogram, you should know it. You should know that the above part of the highest part of the diagram is the vital, the important, the crucial capacity for the lung. The down part is the residual or the left capacity of the lung. However, all of them together is the total capacity. This is all you should know. Okay. So for respiration, we're done. Because we have so much systems and we have to finish the course. So I'm trying to summarize as much as I can and give the most important topics for the Imat. Okay. Perfect. So now, what time is it? Okay, 5 p.m. Okay. Before we continue, see, do we need to know that? No. Start. Can you explain how thickness affecting gas exchange? Okay. So, so the thickness of the respiratory membrane, or the alveolar membrane, let's say, it's important for the gas exchange in what ways? It's important in the effect of the rate of the velocity, the speed of this diffusion to happen. Okay, so the thickness of the alveolar membrane affects the rate of gas diffusion. How? The rate of gas diffusion across this membrane, across the respiratory membrane, is inversely proportional to the thickness. Okay? Rate inversely proportional to the thickness. This means if we have a high thick alveolar membrane, this means that the rate will decrease of diffusion. Why? Because already the membrane is thick. How would the oxygen or the carbon dioxide be able to cross this membrane fastly? However, if it was more thin or low thickness, the rate will increase. Okay? So just this is because of the diffusion of gases like the oxygen, carbon dioxide, and like this respiratory gases, they follow fixed law of the diffusion. Okay. So yeah, like fixed law says that the rate of diffusion is inversely proportional to the thickness, or you can say the distance. Okay, for it to travel. And yeah, so it just speaks about the thickness. You can call the thickness as distance. You can call it surface area. This thing. If it's so thick, the rate of diffusion is low. If it's so thin, the rate of diffusion is faster. And it's logically faster. It would logically be faster. Okay, so now let's end the class. Today we finished respiratory, we finished enzymes. By tomorrow, after tomorrow, we don't have class. Monday, we should be done with hormones, immuno, urinary, reproductive, and nervous. This all of them on Monday. So Monday, we might have like a 4 hours day, like a 4 hours time, or because we should finish the systems. Because after Monday, like Tuesday, we're gonna do genetics, and then Wednesday we're gonna do everything left in chemistry. Thursday, Friday, and then all of the days after we will start solving. So from Thursday we'll start solving. Monday we have to finish the system. Okay, now let's see the questions. Do we need to know oxygen dissociation graph? No. Why is PCl3F2 nonpolar? PCl3F2 Lambert phenomena. Also no. No, these are so detailed, yeah. What's your name? The one asking in direct, what's your name?
Sarthak.
Sarthak.
Yes, yes.
Where are you from?
I'm from India.
You're from India. Okay, from where? Why you're going so deep in studying? You don't really need
Uh,
to know all of it.
yeah, but actually it's in our curriculum book, so I thought maybe it will help.
Yeah, oxygen dissociation graph is not as for the IMAT. It's not even in the syllabus, and the Hamburg also is not in the syllabus. We're working on the syllabus. Okay, they want to
Okay,
bring something
thank you.
out. However, if they brought anything out, it won't be this. Okay, so what was Said? Said was asking something. Yeah, he said PCl3F2 is non-polar. I have to draw it to know why it's non-polar. PCl3 2. Yeah, PCl3F2 is symmetrical. Okay, so yeah, we have here phosphate, chlorine, chlorine. Okay, okay, let me see what he asked. Then why PF3Cl2 polar? Okay, look now because I have to go to work. I really don't have time to wear and go. So I will explain this for you when I reach back from work today. I will send you on private, and if you guys really want, like, if you guys want also, we can send discuss it on group. So if you want, I will draw it for you, both of them. I will explain each one of them on chat on the group. Okay, when I come back from work, because now it might take some time to draw it and things. Perfect. I won't forget you. Don't worry. Okay, so guys, try to practice as much as you can for the weekend. Try as much as you can to try to solve redox reactions, to find oxidation states in chemistry, to balance reactions, to solve things by yourself. Try as much as you can. IMAT is really close to us, and we really need you to succeed. Okay, now yalla,
Uh,
bye and
I want
have
to
a
ask something.
yeah. Who are you?
What Sarthak,
Yeah.
what is the best like material for IMAT? Because my curriculum is very different from what you're teaching right now.
What is the best what?
Like the basic material for IMAT.
Like to solve?
Like no. Which book should I read? Like to get the exact
Book.
idea of the syllabus. Yes, yes.
I sent already a book. I don't know if you're in the group. I sent already a book which is the Oxford book. Let me just make sure which
Oh,
one. To be honest, when I studied for the IMAT, I studied from this book. I will put a point on it in the group. It mainly has
okay, sure.
99% of everything. So me
Okay,
myself, when I studied, I used this book. I will send it now. 1 second. Okay. Yeah, this one. For biology, chemistry, no, not the book, not this book, but for biology, I studied from this. It almost have everything you should know for the IMAT, almost. So if you want, you can use it. Like the notes I'm saying now are from this book, like because I took notes when I
I'm
studied, and
unable
I'm
to
teaching you from
access
this
this
book.
book.
You know? You're not able. Okay, I will send it again. It's so helpful for the IMAT. And the things, the information inside this book are really summarized and like really written in a way that just enter your mind without you reading paragraphs and dying. So it's a kind of helpful book. And the notes I'm teaching you from are from this book. I used to study and summarize each chapter and write notes, and now I'm teaching you from the notes of this. So it might make sense what I'm teaching about, I guess. Okay. So okay. So goodbye and have a good day.